We're damn good too. But you can't be any geek off the street. Gotta be handy with the steel, if you know what I mean. Earn your keep. Regulators! Mount up. It was a clear black night, a clear white moon. Warren G was on the streets trying to console some.
Oh, hey everyone. Thanks for tuning in to the next installment of our dot point 2. We're learning about gene expression, and in this lesson, we're going to look at gene regulation, hence the song. So, what we need to remember before we get started is the transcription process, the translation process, and you'll see here there's a polypeptide gene being produced. That is the end product of gene expression. The gene product is a protein. And that is the case in prokaryotes and eukaryotes. What we need to learn now is that some of those proteins, so some of the genes that code for those proteins, are structural. So there are some genes that code for proteins that actually alter the structure and function of the organism. So structural genes would be genes that code for particular proteins, enzymes, functional proteins, and structural proteins like collagen. So structural genes code for structural proteins. The final product is a protein. But we also have genes that are called regulators. So it's genes that produce proteins that control the action of other genes. And we'll look at examples about what that could mean. But there are some genes that are just the switches for other genes to be switched on or off, like a finger on a light switch. So regulated genes work in two different ways. The one that we're going to focus on is that you have a protein that's coded for by a regulated gene that actually binds to DNA and stops it from being transcribed. So it switches those genes on or off. But we also learned about signaling proteins back in the previous unit. So there's some terminology here we need to be aware of. So when I talk about a regulated gene, I'm talking about a section of DNA that codes for the production of a protein that is a repressor. Now, if you see the word repressor or repressor protein in an exam, that repressor protein is binding. So that repressor protein is binding to part of the DNA and stops that section of DNA from being transcribed. So this regulated gene codes for a repressor protein. The repressor protein binds to a section of DNA that codes for something else, and it stops that section from being transcribed. We've already learned about structural genes, so just sections of DNA that code for a protein or a gene product. And we'll talk about how operator genes work in the next slide. Okay, so before we get started on the example of the lac operon, I want to just highlight why we need to regulate genes and why we need to control which genes are being transcribed when. If you think about it, you've got thousands and thousands of genes on each of your 46 chromosomes, and they don't all need to be transcribed at a particular time. In fact, it would be a huge waste of energy if we did so, and you'd be making products that your cells probably didn't need. we need to be able to control when a protein is going to be made and when a protein needs to stop being made. Many genes should only be activated, or they should only be transcribed when their product is required. And the example I'll go through here is actually from bacteria. So this is the lac operon, and this is what the section of the bacterial DNA looks like. You've got the part of the RNA called the operator, which is like where transcription, of the DNA, called the operator, which is where transcription would start. And then you've got these different segments of the DNA that code for the creation of the enzyme lactase. So bacteria take up the sugar lactose and they need to break it down, and they produce the enzyme lactase to do that. So the default for a bacterium is actually to have a protein, a repressor protein called lac, the lac repressor. The protein actually sticks to that section of DNA, and stops RNA polymerase from binding. So it is not possible to transcribe this gene that is going to create the enzyme lactase. As soon as the bacterium takes up lactose, though, that sugar, which is now in the cell cytosol, will bind to the lac repressor, which means the repressor protein detaches from this promoter site on the DNA. RNA polymerase is able to bind to that section of DNA, and transcription can begin. And that means that once we have transcription to create mRNA, that will then be translated into the protein polypeptide chain, which is in this case going to be lactase. A lot of the exam questions around gene regulation usually aren't very tricky. They look a lot like feedback loops, and they're just asking you to use a bit of logic. So this question is a good example of that. It looks really, really tricky. There's a lot of text. There's a lot of genes you haven't heard of. There's this word antisense regulator that they threw in. But if we think about what the question's actually asking us, it ends up being quite simple. So here it tells us that there's a Hoc gene on the plasmid of a bacterium. So this is the Hoc gene. And it codes for a toxic protein that causes cell death. So the Hoc gene is transcribed to mRNA, and that is then translated to a toxic protein. The Soc gene in the same plasmid codes for an antisense regulator that binds to the Hoc mRNA. Okay, so this gene codes for the Soc mRNA, which actually combines with the Hoc mRNA, and that causes them to be both broken down by an enzyme so that this toxic protein is never made. Why do the bacteria have these genes? I don't know. So looking at the options, it's just asking you how this would play out if the Soc mRNA actually represses or causes the Hoc mRNA to be broken down. So let's look at our options. It would be reasonable to expect that A, Hoc mRNA, would be translated if Soc mRNA was absent. Well, that makes sense. We've got our Hoc gene being transcribed, and if we don't have the Soc mRNA combining with the Hoc mRNA, it will make the toxic protein. So I'm pretty sure that's looking like a good option. Let's just check though. B, Hoc DNA would be unable to be transcribed if Soc mRNA is present. So from what we can see in this diagram and what we've read in the question here is that the Hoc gene is going to be transcribed independently of the Soc gene. So the Soc gene is not controlling transcription here. The Soc gene is actually just producing mRNA to combine with the Hoc gene. So B is incorrect. You would still be able to transcribe the Hoc DNA if the Soc mRNA was present, because that step's clearly able to happen. C, Soc mRNA would have the same nucleotide sequence as Hoc mRNA. Well, that's definitely wrong, because otherwise they would be coding for the same toxic protein, and that's clearly not the case. And so A is still looking like a good answer, but we'll just finish off with D. An E. coli cell without any plasmids would be killed by the toxic protein. No, because the toxic protein is coded on a plasmid. So obviously if the E. coli cell didn't have that particular plasmid, it would not create the toxic protein and it would probably be fine. So what I've done in this question is I've actually used the diagram, logic, and my understanding of how a repressor could work to pretty much use logic and diagrams to come to the conclusion of A. I didn't have to know what Hoc was, I didn't have to know what Soc was, and even though that question looked really hard, it was all right. Same thing goes for this one. Now this is on page 20 of the exam booklet that I gave you yesterday. And you'll see that you first have to explain what's meant by gene regulation, and hopefully the previous slides have allowed you to write that gene regulation is the switching on or off of transcription of genes when required. You've probably got a better definition you could put in there, and please check the examiner's report to make sure you're happy with it. But let's look below. So we're given this text and a diagram. Again, it's looking really tricky. In summary, it's just explaining that bacteria need to produce proteins and they need amino acids from the food they've digested, from the food humans have digested in the gut bacteria to do that. If your diet isn't quite right, your gut bacteria are not going to be able to obtain the amino acids they need to make the protein. But that's okay because they can make the amino acids themselves. And we actually see that pathway below. So this is a regulation system in a bacterial cell, and it is a pathway to creating the amino acid tryptophan. So if the bacterium isn't able to get tryptophan from the diet, hopefully this pathway would be triggered to create that amino acid so the bacterium could survive. Okay, it says tryptophan is the regulatory compound in these two pathways and acts as a repressor in both. This is a really important part. It's telling us here that in the presence of tryptophan, regulation pathway X will be repressed. And it's also telling us that tryptophan is a repressor for regulation pathway Y, which means tryptophan will prevent regulation pathway Y from occurring whenever it's there. So if you look at the question here, describe the immediate outcome when tryptophan activates pathway X. If we read in the question that tryptophan is a repressor, and we see here that pathway at X will repress all of this stuff, we should be able to describe the immediate outcome pretty easily. The trick is realizing it's a repressor. So the outcome would be that the transcription of genes 1 through 5 would not occur. These genes would not be transcribed. You got it? To check if you do, please continue on with these questions on page 20. It will ask you, what is the effect if tryptophan is present on pathway X? And remember, it's a repressor. So pause the video and give that a go now. Okay, so the final part of this dot point is actually learning about mutations. So mutations, which we learned in year 10, are just changes in the genetic code. And we're going to learn a little bit about how these mutations occur, but more importantly, what these mutations are and how they can throw out the codons and create really dysfunctional proteins. Okay, so we need to realize that mutations, so changes in the genetic code, they happen spontaneously. They happen through errors in DNA replication. They happen sometimes through environmental factors like exposure to radiation or carcinogens. And really, most mutations are going to be harmful, but there are many examples in evolution, which we'll learn about, where mutations can be useful. So an example of a mutation that's harmful is in cystic fibrosis. Just one incorrect amino acid that results from a mutation in the gene that creates the protein for the mucus that clears out our lungs means that cystic fibrosis patients have this sticky mucus and they have mucus filling their lungs for their whole lives. So that's an example of a really harmful mutation. A useful mutation, the best example we have in human evolution is the quite recent mutation where our ancestors who all had brown eyes, they started to move to colder climates with less light availability. And there was a spontaneous mutation which prevented the brown pigment in the eyes from forming, leaving blue eyes. And the blue eyes were able to absorb more light and enable our very, very distant ancestors who lived in the north of Europe to survive much better than their brown-eyed counterparts. So that's an example of a useful mutation. You can also imagine a polar bear being born with white fur for the first time, being able to blend into the snow, unlike its brown bear counterparts. So yeah. We'll talk about how natural selection promotes these beneficial mutations and kills off harmful mutations. But we'll talk about where they come from. Okay. So you will have spontaneous mutations occurring in transcription and from radiation. We know that some of the mutations will occur in germline cells, so gametes, and that means the mutation will be passed on to the next generation. If it occurs in a somatic cell, that might disrupt your structural function, but you will not be able to pass it on because your somatic cells don't go through meiosis. Okay, so chromosomal mutations just refer to the fact that there's really big chunks of DNA that are abnormal. And we learned about what can happen during non-disjunction of chromosomes, resulting in polyploidy or aneuploidy, or even whole pieces of chromosomes missing or being duplicated in translocation. What I want to focus on now are point mutations. So where there's a change to only one or two bases, and these mutations can have a significant impact on protein function. Okay, we have to know what all these different types of point mutation are. And a point mutation is just where a single base in the genetic code is changed. So I'll go through examples of these and what the effect is on gene expression. So the first type of mutation is substitution. So that's just where one nucleotide is replaced with another. So if you have the sequence CGC, one of the letters is substituted, so CGC, the third C is substituted to a T, so CGC changes to CGT. And this can sometimes result in dysfunction, but it still keeps the same number of bases, so 3 bases before and 3 bases after, so it's not as bad as some of the others. The next one is inversion, so that's just where 2 nucleotides are switching places or being reversed. So CGC would be inverted to CCG, for example. The ones that cause the most damage are insertion and deletion. So insertion is just where there's an extra base or 2 added into the DNA sequence. So CGC would be CGCCC. That's an example of an insertion. And deletion is where you actually lose a base. So maybe it hasn't been replicated or transcribed properly, and so CGC would be CG. These last two, insertion and deletion, can cause something called a frame shift mutation. And frame shift mutations are particularly bad because you're changing the number of bases in that genetic code, which actually throws out the codons. So remember that codons code for an amino acid. So for example, this amino acid serine is coded for by UCU, which comes from the DNA combination AGA. And histidine here is coded for by GTA. If you take out or add in a base through insertion or deletion, you're actually going to change all of the codons after it. So let's see what's happened here. We should be having GTA producing histidine. But unfortunately, there's been a deletion. We've got GT, the A's been deleted, and we've got the C being squished in there now. So the codon is now GTC. And the next codon, instead of being CTA, is now TAG, and so on. So the effect of an insertion or deletion is it actually throws out every subsequent codon and therefore can change every subsequent amino acid which means that you would probably end up with a protein that either doesn't work or it has a stop codon far too early, and that's where you get the serious dysfunction. So, as I said before, some mutations might not have any effect on the protein that's produced. So, for example, if you get a very simple substitution, the original strand was TAT, and the resulting shift was ATA, so you get a change in the code. That will still result in UAU, which will still code for the same tyrosine codon. Okay, so just to be clear, if the gene is TAC, and that codes for a tyrosine, which is what's needed, a change here, so TAC going to TAT, that will still produce a tyrosine, and so that mutation is silent. We can't see the effect of that mutation because, fortunately, TAT codes for tyrosine just like TAC. But if the mutation is to a TAG that doesn't code for tyrosine, it actually codes for a stop codon. And so that would get an incomplete protein. And that's because we had a different codon and therefore a different amino acid. That's because we had a stop codon. And finally, if you have another type of point mutation, and this codes for a missense mutation, so we see that it's creating a different sequence of amino acids. That's going to result in a protein with the incorrect amino acids. Maybe it will fold in the wrong place, maybe it won't fold in at all, and it won't be able to perform its function. And finally, just to remember that you have to identify, sometimes in a question, whether the mutation is somatic, so in a somatic cell which cannot be inherited, or a germline mutation. And the picture I have here is an example of a mother, and she has given birth to a child with albinism. The mutation that causes albinism must have occurred in either her or the father's gametes, so it could be passed on to the next generation. Somatic mutations would only affect the person that they're occurring in. Okay. I've got a question here that I'd like to finish off on. It's just asking us what type of mutation has occurred here. So it says that there's a disease, Tay-Sachs, that is caused by mutations in the hexoceaminidase gene, resulting in an abnormal enzyme. And so we've got the genetic sequence for the normal enzyme. So we should be seeing these amino acids, which are coded for by these codon sequences of RNA. Something's happened here where we now have the incorrect RNA codons coding for the wrong amino acids. So instead of serine, we're getting tyrosine. Instead of valine, we're getting serine, and so on. All this question is asking is, what happened? where did the mutation occur and what type of mutation was it? So if we look at what's been proposed, A says there's been a substitution of G for C at the beginning of codon 7. I think we can rule this out because not only do we have issues at codon 7, so we've got the wrong amino acid at codon 7, codon 6 and codon 5 are incorrect. So I wouldn't blame a substitution at the beginning of codon 7 for that. Furthermore, that's not a substitution. Second, B, a duplication of ACU at the beginning of codon 4. Well, if it was a duplication of ACU, we should see ACU, ACU, and we don't, so rule that out. Next, an insertion of UAC at the beginning of codon 5. Let's take a look. So ACU, ACU, codon 5. Yep, so it looks like UAC is definitely present in codon 5. The way to check that it's insertion is we should see what was meant to be in codon 5 in the next one. And yes, we can see that there. And that has thrown out what was in codon position 6 into codon position 7 as well. So C is looking like a really good option, but just to rule out D, deletion of UCU at the beginning of codon 6. No, because codon 6 shows UCU, and we can see here that it's just been pushed back. So rule out D, and the answer is C, an insertion of UAC. If you found that pretty easy, I think we're in an excellent position to keep going next lesson. Thank you so much for spending your time learning biology. Okay, thank you.